Okay, assalamu alaikum everyone. So yesterday we were studying levels of protein folding and we understood that how is the primary structure, the secondary structure and the tertiary structure is formed. Now, so far the three types of or the three levels of protein folding that we studied, they were basically concerned with what? They were concerned with one single polypeptide chain. With that, I mean that when you were talking about the primary structure, we were saying that the sequence of amino acid in a polypeptide chain. So the number or the type of amino acids and the sequence of the amino acids in a polypeptide chain, how they line up, how they bond and form peptide bonds between them, that basic fundamental or that basic foundation of a protein molecule, that is known as your primary structure. Then, when you were talking about the secondary structure, we understood that the adjacent or the amino acids within one specific or one polypeptide chain, they actually have what? They have C double bond O and NH groups. And we understood that when hydrogen and oxygen interact via weak interactions, that type of bonding is what? That is hydrogen bonding. So when the C double bond O oxygen of one amino acid and the hydrogen of the amino group form weak links between them. What happens is that the polypeptide chain bends over itself. So either it forms a spring like helical structure, which is known as the alpha helix, or a much rather flexible structure, which is your beta pleated sheet. So we denote a beta pleated sheet with arrows or with ribbon like structure. And the alpha helix basically looks like what? It looks like a spring or a DNA molecule, something like this. So you see, there is one particular polypeptide chain which is being wrapped upon itself or which is being bended upon itself. So that type of a structure is a secondary structure where we form alpha helixes and beta pleated sheet. So far, the primary and the secondary structure were concerned with the peptide bonds. हमारा peptide bond क्या होता है? हमारा peptide bond is basically the C double bond O NH bond, right? But when we talk about the tertiary structure of an amino of a protein or a polypeptide chain, we are concerned with the R groups. Now, R groups for all amino acids can vary, right? Every amino acid has a unique R group, and we studied the R group of two specific amino acids, which were glycine and cysteine. So for glycine, we said that the R group of this particular amino acid is what? It's just a simple hydrogen atom. So that is basically the structure of a glycine molecule, which you should know for your syllabus. And then we studied about cysteine. We'll come to cysteine in just a bit. But what exactly is tertiary structure? What happens is that the R groups, they're left, अभी तक उनकी कोई bonding, उनकी कोई interaction हमने देखी नहीं. अभी तक जो भी secondary structure और primary structure में bonds हो रहे थे, they were concerned with the C double bond O N structure. But the R groups are basically not interacting as of yet, लेकिन हर amino acid पे कोई ना कोई R group तो होता है. And we understood that there are three types of R groups. Either they are polar, which means that they have a partial positive charge on them. And then charged R groups, we said that they have a complete charge on them. And we said that because water is a polar molecule with this partially positive, partially negative oxygen and a partially positive hydrogen, because of these partial charges present in a water molecule, we identify water as a polar molecule. And whenever any charged species, be it a polar charged species or a completely charged species, when it comes near the vicinity of a water molecule, water forms hydrogen bonding with them, right? Or water forms weak interactions with them. और वो water soluble भी होता है. So then we understood that if two polar R groups are adjacent to one another, यानी दो amino acids, एक हमारे पास पूरी polypeptide chain है, यहाँ पर एक amino acid होगा. Like there would be multiple amino acid, but what we are concerned with are the two amino acids which are facing each other. So the two amino acids which are facing each other, if they have they have polar R groups. So the interaction between polar R groups is what? It's a type of hydrogen bonding. Then if two non-polar R groups are face to face with one another, then they would interact via hydrophobic interactions. Why? Because non-polar है तो non-polar में कोई partial charge या कोई permanent charge होगा नहीं, जिसकी वजह से they would be moving away from water, and because they have this common link, they have this common mutual factor that both of them are water heating groups, they would form hydrophobic interactions. Then for the charged R groups, if a positively and a negatively charged R group is face to face, they would form an ionic bond. Now the fourth type of linkage, or the fourth type of bond that we studied, was a disulfide linkage. And a disulfide linkage is formed when two cysteine molecules are present together, or they're present face to face, or they're present adjacent to one another. So what happens is that this is basically the R group which is present on a cysteine amino acid, which is CH2SH. And what happens is when the two of them are face to face one another, two hydrogen atoms are lost, and this sulfur bonds to the sulfur of the other cysteine molecule. And because two sulfur molecules are bonding together, this type of a linkage is called a disulfide linkage. And amongst all these three, disulfide linkage is a very strong type of bonding. So tertiary structure basically एक ही polypeptide chain में हमारे पास जो adjacent amino acids के R groups होते हैं, उनकी किस तरह से interaction हो रही होती है, उसकी basically folding के level को कहते हैं. So this is what we've studied so far. Any questions the two of you might have regarding anything that we've studied? Or is this clear? Okay, perfect. So let me show you the tertiary structure. How does it look like? So this is what a tertiary structure look like. Now you can see यहाँ पर हमारे पास अल्फा हेलिक्सेस भी हो सकता है, बीटा पीटेड शीट भी हो सकता है, बट ये एक ही पॉलीपेप्टाइड चेन है जो इस तरह से आपस में फोल्ड हो गई है. फोल्ड क्यों हो गई है? बिकॉज ऑब्वियसली अलग अलग तरीकों की इंटरेक्शन हो रही होंगी, जिसकी वजह से जो बॉन्ड्स हो रहे होंगे उसकी वजह से हमारी जो पॉलीपेप्टाइड चेन है इट इज बेंडिंग अपॉन इटसेल्फ. तो हमारी देखो, ये हमारे पास बीटा प्लीटेड शीट थी, ये हमारे पास अल्फा हेलिक्स था. So if you compare the alpha helical structure and the beta pleated sheet structure, तो यहाँ पर और यहाँ पर, if you could compare these two, the secondary structure with the tertiary structure over here, can you appreciate and observe कि tertiary structure में level of folding, level of bending upon itself ज़्यादा increase हो गया है? यानी tertiary structure is far more folded, it is far more bended than an alpha helix or a beta pleated sheet. Can you observe that? Okay, so this basically is what one level increase of a protein folding. ठीक है जी. Now, so far, the three types of foldings that we studied were concerned with one single polypeptide chain. यानी एक ही polypeptide chain आपस में किस तरह से interact कर रही है, किस तरह से bond कर रही है, किस तरह से fold कर रही है, उस interaction को या उस तरह की folding को हम discuss कर रहे थे. Now, let's move to the fourth and the highest level of protein folding, which is a quaternary structure. And अभी हम उसको बिल्कुल discuss करेंगे. So what exactly is a quaternary structure? Now over here in quaternary structure, when more than one, यानी कि 2, 3, 4, 5, 6 polypeptide chains come face to face on another, उनके बीच में किस तरह से bonding होती है, we would discuss that. So first let me write the definition for this. So quaternary structure forms when two or more than two polypeptide chains associate via either of the following four bonds. और वो किस तरह के following four bonds हैं? वो exactly वही four bonds हैं जिनको अभी हमने ऊपर discuss किया है. Between, अच्छा ये मैं एक fill in the blank छोड़ रही हूँ. I want you guys to tell me कि ये जो bonds हैं वो किसके बीच में बन रहा है? Of amino acids between neighboring polypeptide chains. अच्छा, तो हमारे पास जो ये bonds हो रहे हैं, ये बॉन्ड्स हमारे पास आर ग्रुप्स की वजह से हो रहे हैं जो कि अमीनो एसिड्स पे प्रेजेंट है, या फिर ये हमारे पास जो पेप्टाइड बॉन्ड्स हैं, सी डबल बॉन्ड ओ एन, उनकी वजह से हमारे पास ये क्वाटरनरी स्ट्रक्चर वाली फोल्डिंग हो रही है? व्हाट डू यू थिंक? इज इट बिकॉज ऑफ द प्रेजेंस ऑफ द आर ग्रुप्स? Are the R groups of the neighboring polypeptide chains interacting, or the C double bond O, which is your C double bond ONH, क्या ये वाला बॉन्ड इन्वॉल्व है इस तरह की बॉन्डिंग में, या हमारे पास R ग्रुप्स इन्वॉल्व है? व्हाट डू यू थिंक? श्योर। सो आई वाज सेइंग कि हमारे पास क्वार्टनरी स्ट्रक्चर में क्या चीज इन्वॉल्व हो रही है? हमारे पास क्वार्टनरी स्ट्रक्चर इज व्हेन नेबरिंग पॉलीपेप्टाइड चेन्स, जो या तो वो फेस टू फेस होंगी या वो बराबर बराबर होंगी, बट व्हेन मोर देन वन, टू, थ्री, फोर, फाइव, सिक्स, व्हेन डिफरेंट पॉलीपेप्टाइड चेन्स दे इंटरैक्ट, व्हाट बॉन्ड, यानी इस तरह के क्वार्टरनरी स्ट्रक्चर में जब हमारे पास बॉन्डिंग हो रही होती है, तो वो हमारे पास बॉन्डिंग पेप्टाइड बॉन्ड्स की वजह से हो रही होती है, एडजेसेंट पेप्टाइड बॉन्ड्स की वजह से नेबरिंग पॉलीपेप्टाइड चेन्स लिंक टुगेदर करते हैं, या जो आर ग्रुप्स होते हैं हमारे अमीनो एसिड्स के, जो कि पॉलीपेप्टाइड चेन्स में होते हैं, आर ग्रुप्स की वजह से ये बॉन्डिंग हो रही है? व्हाट डू यू थिंक? इज द पेप्टाइड बॉन्ड इन्वॉल्व इन द क्वार्टरनरी स्ट्रक्चर, or the R groups of the amino acids present in the polypeptide chains, are they involved? What do you guys think? Okay, so Hajra is saying both. Abhiya, what do you think? Okay, now let me draw a primary structure in front of you guys, or let me draw the complete thing. C double bond O NH, ठीक है? C double bond O NH, and let me draw the polypeptide over here. And let me copy this. Okay, so ये for example इसका R group है. Let me draw it with another color. ये हमारे पास R groups हैं, R groups हैं, R groups. Now, over here हमारे पास जो पेप्टाइड बॉन्ड होता है, उस पेप्टाइड बॉन्ड का क्या रोल होता है? उस पेप्टाइड बॉन्ड का रोल ये होता है कि वो एक कनेक्टिंग लिंक होता है बिटवीन टू अमीनो एसिड्स, ठीक है? उसके बाद उसका जो दूसरा रोल होता है वो ये होता है that it basically forms the alpha helix and the beta plated sheet because of hydrogen bonding. Now what happens is कि हमारे पास जो R groups होते हैं of our amino acids, वो बेसिकली हमारे पास जो फर्दर फोल्डिंग होती है वो कराते हैं. दैट इज, यू रिमेंबर दिस, कि हमारे पास जो फर्स्ट टू स्ट्रक्चर्स हैं, यानी प्राइमरी एंड सेकेंडरी, उसमें हमारे पास हमारे पेप्टाइड बॉन्ड्स इन्वॉल्व होते हैं, और जो हमारे पास टर्शरी और क्वार्टिनरी स्ट्रक्चर होता है उसमें हमारे पास कौन से ग्रुप इन्वॉल्व होता है? उसमें हमारे पास आर ग्रुप्स इन्वॉल्व होते हैं. सो दैट इज हाउ आई रिमेंबर that the first two levels of protein folding, they are concerned with the peptide bond, and the last two um, levels of protein folding, they are concerned with the R groups. And just in a second, I'll explain why is it so. Now, हमारे पास जो 4 bonds हमने study किए थे वो कौन कौन से थे? Hydrogen bonds थे. Then we studied hydrophobic interactions. And then we said that there are ionic bonds. And then we said there are disulfide linkages. अब अगर मैं बात करूं हमारे पेप्टाइड बॉन्ड की, तो मेरा पेप्टाइड बॉन्ड आपस में सिर्फ एक ही तरह की बॉन्डिंग कर सकते हैं. वो किस तरह की बॉन्डिंग होगी? हाइड्रोजन बॉन्डिंग होगी, राइट? और यहाँ पर हमारे पास जब क्वार्टरनरी स्ट्रक्चर फॉर्म हो रहा होता है, तो ये चारों लेवल्स ऑफ चारों टाइप्स ऑफ बॉन्ड्स वी आर ऑब्जर्विंग. एंड वी नो कि हमारे पास हाइड्रोजन बॉन्ड्स तब बनते हैं जब हमारे पास पोलर आर ग्रुप्स इंटरैक्ट कर रहे हो. हाइड्रोफोबिक इंटरैक्शन कब होती है? जब नॉन पोलर इंटरैक्ट कर रहे हो. आयोनिक इज व्हेन चार्ज्ड स्पीशीज आर इन फ्रंट ऑफ ईच अदर. And disulfide occurs when there are two cysteine molecules facing or adjacent to one another. Now, हमारे पास if you notice over here, जो quaternary structure होता है उसके लिए हमें कम से कम 2 प्रोटीन पॉलीपेप्टाइड चेन्स चाहिए होती है. अब वो पॉलीपेप्टाइड चेन्स सेम भी हो सकती हैं, वो पॉलीपेप्टाइड चेन्स डिफरेंट भी हो सकती हैं। सेम मतलब हमारे पास फॉर एग्जांपल इफ आई हैव एन अल्फा ग्लोबिन, और अभी मैं बताती हूँ अल्फा ग्लोबिन क्या है। तो मेरे पास अगर एक अल्फा ग्लोबिन और मेरे पास दो अल्फा ग्लोबिन हुई और ये आपस में इंटरैक्ट कर रही हैं, तो होगा क्या कि इफ देयर आर टू और मोर देन टू polypeptide chains interacting, that is when our quaternary structure is formed. Our primary, secondary, and tertiary structure, we are only talking about one polypeptide chain. But if two or more than two polypeptide chains are face to face one another, their R groups would form linkages, their R groups would form bonds, and they can either be the hydrogen bond, the hydrophobic interaction, the ionic bond, or the डाइसल्फाइड बॉन्ड, और एक से ज्यादा भी हो सकते हैं। फॉर एग्जाम्पल अगर मेरे पास ये वाला आर ग्रुप है, तो ये मेरे पास पोलर है, ये मेरे पास पोलर है, तो इनके बीच में कौन सी बॉन्डिंग होगी? व्हाट डू यू थिंक? व्हाट टाइप ऑफ अ बॉन्ड वुड बी बिटवीन टू पोलर ग्रुप्स? What type of a bond would be present if two polar groups are face to face or adjacent to one another? Yes, Hajra, you're right. So this is a type of hydrogen bond. And subsequently, ये मेरे पास एक cysteine amino acid है, ये मेरे पास cysteine amino acid है. We know when two cysteine amino acids, their R groups interact, they form disulfide linkages, right? And then for example, यहाँ पर मेरे पास again hydrogen bonding है, यहाँ पर for example मेरी hydrophobic interaction है. तो किसी भी किस्म की इन चार में से बॉन्डिंग हमारे पास हो सकती है quaternary structure में. Now, for example, let me write three different proteins and let me tell you how many polypeptide chains are present in them. So first of all, let's talk about a very famous and a very known protein, which is your hemoglobin. So hemoglobin is made up of four polypeptide chains. Then we have insulin. It is made up of two polypeptide chains. And then we have myoglobin. This is basically a protein present in your muscle cells. It is made up of one polypeptide chain. Now tell me which of these would be having a quaternary structure and which of these would be having just a tertiary structure. Which of these would be having just tertiary structure and which of these can possess quaternary structure as well, and why? Okay, so hemoglobin will have a quaternary structure. That's correct. Hemoglobin can possess a quaternary structure. What about insulin? Okay, so you're saying insulin would possess a tertiary structure. Yeah, what do you think? Why would insulin? Okay, so Hajra is saying insulin will have a secondary structure, and Abiha is saying that it would have a tertiary structure. I want the two of you to give me explanation for both of your answers. Why do you think that insulin is having tertiary for Abiha, and for Hajra you said that it's secondary? So I want you guys to give a reason for your answer. Because it has two polypeptide chains connected by a disulfide bond. Okay, Abiha, why do you think that there's a disulfide bond? Because disulfide bond तो खाली cysteine amino acids के दरमियान होता है, and we haven't specified कि insulin में cysteine होती है कि नहीं. And Hajra, what about you? Acha, because it has two alpha chains and two beta chains. That is what you were saying for insulin. Acha. So when we're talking about a quaternary structure, any protein which has two or more than two polypeptide chains, या तो उसके पास दो polypeptide chains हो या दो से ज्यादा polypeptide chains हो, it would have a quaternary structure. And why is that so? क्योंकि हमारे पास primary structure, secondary structure or tertiary structure, these types of structures are formed in one single polypeptide chain. एक single polypeptide chain की जब बात हो रही होती है, उसकी फोल्डिंग की बात हो रही होती है, तब हमारे पास जो लेवल ऑफ फोल्डिंग होती है वो फर्स्ट लेवल क्या होता है, सीक्वेंस ऑफ अमीनो एसिड्स व्हिच वुड फॉर्म द बेसिक फ्रेमवर्क ऑफ दैट पॉलीपेप्टाइड चेन. Then we have alpha helixes and beta pleated chain, which is basically the folding of the polypeptide chain onto its own self. यानी वो अपने ही अराउंड या तो रैप हो रही है या वो बेंड हो रही है. Then tertiary structure में उसी polypeptide chains के जो R groups हैं वो आपस में interact करके further fold हो रहे हैं. ठीक है, if you could notice टर्शरी स्ट्रक्चर में खाली एक यूनिट दिखाया, खाली एक कलर से उन्होंने एक पॉलीपेप्टाइड चेन शो किया. सो दिस इज वन सिंगल पॉलीपेप्टाइड चेन, राइट? बट व्हेन देयर आर टू पॉलीपेप्टाइड चेन्स और मोर देन, फॉर एग्जांपल यहाँ पर मैं कोई थर्ड ड्रॉ कर दूं तो यहाँ पर ये मेरा सब यूनिट जो हो गया वो थर्ड हो गया. So whenever more than one polypeptide chains are present, they would interact because they have their R groups. उनके R groups जो होंगे वो आपस में interact करेंगे दूसरी polypeptide chain के साथ, and they would interact by either hydrogen bonding, hydrophobic interactions, disulfide linkages, or ionic bonding. So if there are two or more than two polypeptide chains, be it the same polypeptide chains or different polypeptide chains, अगर दो से ज्यादा या दो chains present हैं, so it would always form a quaternary structure. Now tell me, what do you think? Would insulin be having a primary, secondary, tertiary, or quaternary structure? What would be your answer? Insulin is made up of two polypeptide chains, right? So if it is made up of two polypeptide chains, so for example, उसकी ये एक chain है, and then there is this another chain. So when there are more than one chain present in front of each other, they would be interacting by either of the four bonds that we have studied previously, and therefore, as Hajra mentioned, insulin would be having a quaternary structure. Now, myoglobin. What do you think? Would myoglobin possess a quaternary structure, or would it just have a tertiary structure or a secondary structure? What do you think? Okay, what about you, Abiha? What do you think? What would be the structure of myoglobin? Okay, so you both are saying that myoglobin would have a primary structure. Okay, so okay, okay. So basically हमारे पास myoglobin जो है उसमें हमारे पास खाली एक polypeptide chain है. So we know that quaternary structure cannot be formed over here. Why? Because for quaternary structure we need at least two polypeptide chains. So we know that myoglobin cannot have a quaternary structure. Now, for myoglobin, it can both have a secondary and a tertiary structure. Why? Because हमारे पास जो ये levels of protein foldings है ना, it's like okay, सबसे पहले जब assemble हुआ हमारा polypeptide chain, तो ये level of protein folding हुई. फिर उसके बाद अगर उन adjacent amino acids ने एक secondary structure बनाया, तो उन्होंने secondary structure भी formulate कर लिया. And then once the secondary structure, which can either be an alpha helix or a beta pleated sheet, if those secondary structures interact further and they're folded by the linkages of their R groups, they would form tertiary structure. So basically, think of these levels of protein folding as a pyramid, as a hierarchy. तो हमारे पास सबसे पहले क्या होता है, हमारा primary structure, then another level up we go, that's secondary, then we form tertiary, and then we have quaternary. So हमारे पास we know कि जो myoglobin है उसके पास ये नहीं हो सकता, बट इन तीनों में से कोई भी स्ट्रक्चर मायोग्लोबिन का पॉसिबल है. वी जस्ट हैव टू नो दैट द क्वार्टनरी स्ट्रक्चर इज नॉट पॉसिबल. अब उसमें कितनी लेवल्स ऑफ प्रोटीन फोल्डिंग्स हुई हुई हैं वो इन तीनों में से पॉसिबल होता है. यूजुअली द हाईएस्ट लेवल and the maximum level of protein folding which we can observe in a myoglobin molecule is tertiary. But because it is only a one single polypeptide chain protein, सिर्फ एक ही पॉलीपेप्टाइड चेन से बना हुआ वो प्रोटीन है, उसमें कोऑर्डिनरी स्ट्रक्चर वी कैन नेवर ऑब्जर्व. ये तीनों स्ट्रक्चर्स आर ऑब्जर्वेबल इन मायोग्लोबिन मॉलिक्यूल्स. ठीक है, एंड अगर इसकी रीजनिंग आ गई, सो यू वुड से दैट मायोग्लोबिन कैन नॉट हैव अ कोऑर्डिनरी स्ट्रक्चर बिकॉज इट हैज ओनली वन पॉलीपेप्टाइड चेन. ठीक है, सो इज दिस क्लियर? हमारे पास प्रोटीन फोल्डिंग आप लोगों को चार लेवल्स ऑफ प्रोटीन फोल्डिंग आर दे क्लियर?